Good morning. Welcome to Psych 9A. Why do I have such a large smile on my face? Does anybody know? At the end of class today, we will be 100% complete as far as class meetings go. That's why I'm so happy. We, of course, have a final exam next week on Tuesday, but I'll talk about that towards the end of class today. So class today actually has two parts. It'll be completing Chapter 6 first, followed by talking about the final exam second. If there are no further questions, I want to jump right in, because I know at the end of this class we're 100% of the way done. How do people feel about that? Start right up? Good, let's start right up. Finishing Chapter 6 on consciousness, well, there were 4 topics in the later part of this chapter remaining for us to discuss, including sleep and dreams. And we'll be talking a fair amount about EEG signals, interestingly enough. We'll also talk briefly about hypnosis, meditation, and drug-induced changes. So, sleep and dreams. You're probably familiar with the fact that we sort of have this cycle or rhythm with which we lead our lives. We're awake for some period of time and then we go to sleep. And this is fairly regular. It turns out that the natural cycle, a diurnal cycle, is actually a little bit longer than 24 hours. For most people, it's closer to 25 hours. In any event, what happens every 24 hours typically is that, well, our alertness will increase towards the later part of the awake part of our day, and then it will decrease, and then we'll go to sleep. Then the next day, something similar will happen. Alertness will increase throughout the day and then go back down, and we fall asleep. If we look at body temperature, there's a similar trend. When we wake up, the body temperature rises. And towards the end of the day, you may or may not see an increase, but certainly by the time you're back asleep, it's down again. Back up during the day, down again during sleep. And interestingly, particularly I think for younger people, growth hormone, there's a little bit of it secreted during the day, but it's primarily at night when one is asleep that you get this secretion of growth hormone. So if you know kids who want to grow just a little bit taller and bigger, well, make certain that they get adequate sleep, because that is when growth hormone is generated. So cells in the hypothalamus control the brain arousal level, and that includes the circadian rhythms. And the hormone melatonin mediates the sleep-wake cycle, which is also influenced by visual input. I believe I mentioned briefly retinal ganglion cells that are specialized for signaling light levels, day versus nighttime light levels. The timing of one's meals, cortical activity, work that one has to do with the brain, for example, staying up late to do homework, that sort of thing also has an influence. So, if we now examine brain activity in a little bit more detail using the EEG method, electroencephalography, then we see that there are distinct stages during the sleep cycle. And people typically identify 4 of these. And note that if one is awake, then there's typically a lot of EEG energy in the beta frequency range. So if you look at the EEG waveform, say, collected from an electrode at the top of the head, and you examine how they're jiggling along, well, beta rhythms are effectively referring to energy in the maybe 14 through 30 hertz range. So let's pick an example in that range, maybe 20 hertz. So we'll have 20 cycles per second for that particular beta range frequency. Anyway, if one is alert and wakeful and thinking and moving around, then there's typically a lot of beta energy. This diminishes as one becomes more relaxed, and in particular if you're sitting back and closing your eyes and going like this, well, that beta energy will decrease and alpha wave energy will increase. Now the alpha waves again are more or less between say 8 and 12 hertz, and the standard example would be 10 hertz. So you have 10 full cycles of that wave per second. And this is associated with closing your eyes, relaxation, And, well, right before you go to sleep, you'll see a lot of alpha wave energy. So, what happens then is that you will fall into sleep and exhibit successively stage 1, 2, 3, and 4 type waveforms. Theta waves, I mentioned these very briefly. Theta refers to the frequency range 3 hertz through 8 hertz. So what's a good example of that? Say 5 hertz. So if you have a wave that's happening 5 times a second, that would be a theta band wave. And it's frequently the case that when you're awake, you can see theta waves in your frontal lobes. But if you have just begun to fall asleep, if you're in a light sleep, then you'll also see theta waves fairly generally across the surface of the scalp. And that's in the, say, 4 through 8 hertz range. These are also observed during rapid eye movement sleep, which I will talk about in just a sec. Stage 2, a little bit deeper, you will see that we still have some fairly high frequency waves. We also have what are called K-complex spikes. Stage 3 and stage 4, these are associated with deep sleep. And you can see in stage 4 that we have these very slow waves. These are, maybe 1 or 2 hertz. So these are delta waves. We have sort of a mix of various frequencies of delta waves here. And as we ascend out of sleep, well, we have higher and higher frequencies of waves present. So let's see what this looks like a little bit more systematically. If we're awake, then we're up here. Not in a sleep stage, but as soon as we fall asleep, there'll be a brief period during which EEG will show these stage 1 theta waves. That will be followed by stage 2 type waves in which you'll see sleep spindles and K-complexes. Then stages 3 and 4 are deep sleep where you'll see these delta waves. And this will last, well, this is a timeline here in hours. And this will last some period of time, the deep sleep. Then you'll start coming out of it, and once you reach stage 1 once again, you may have rapid eye movement. Rapid eye movements occur, and I'm certain that you've noticed either in somebody you know or maybe in one of your pets, sometimes when it's sleeping, you can see the eyes moving underneath the eyelids. Have you ever noticed that in a friend or a cat or a dog or something? Yeah. So these are periods during which that person or the animal is dreaming. It's that simple. And they are making rapid eye movements, and you can't really see the eyes move unless you lift up their eyelids, but you sometimes see the little eyelid jiggle a little bit. And that's when you know they're dreaming. If you have a pet dog or a pet cat, sometimes you can see them move their paws a little bit, as though they're chasing an animal in their dreams. So that's this period here marked in red. That lasts a short while. And then again, during a normal sleep, one will descend into deep sleep, down into stage 4, come back out again. And you can see over the course of a single night's sleep, there will be several periods during which dreams or rapid eye movement sleep occurs. And there will also be several periods during which we have this sort of slow wave deep sleep. And eventually, after however many hours, in this case it looks like approximately 7.5, the person wakes up. So it's interesting, and I don't know if you've ever noticed this, that skeletal muscles are paralyzed while one sleeps, particularly during the rapid eye movement phase. And has anybody ever woken up while you've had sleep paralysis? Yeah, it's sort of weird. That happened to me once, and I was sort of freaked out because I knew I was awake, but I couldn't move my body, and it's actually a little bit scary. But your body is normally, it sort of paralyzes itself during the rapid eye movement sleep, so you don't start running around and doing all sorts of crazy things in your bed. So you're sitting there, more or less sleeping, paralyzed, to protect against that sort of thing happening. There's another interesting thing. How many people, when they're falling asleep, have ever had one of their muscles, or maybe a group of muscles, sort of have a little spasm, like, anybody ever have one of those when they're falling asleep? Yeah, that's called a myoclonic seizure. And it's effectively a number of motor neurons acting together. This is typically right before you fall asleep, and they might jerk the whole side of your body or something like that. And it's a key indicator that you're just about to fall asleep. So even though it can be disturbing, it's usually a good sign, yes, you're just about to fall asleep. So bear with it. Okay. So turns out that people need to have adequate amounts of both deep sleep and rapid eye movement sleep. And it turns out that if you are deprived of sleep, well, that's not a good thing. And this is why I try to remind you, when it comes to taking exams, you don't want to stay up the night before or do any of this cramming business. What you want to do is study early and often, so when it comes to final exam week, you actually get nights where you have good sleep. Because what does sleep deprivation lead to? Well, let's see. Loss of alertness, feeling bad. Who wants to go around feeling bad? Poor task performance. Yikes. So here is a critical thinking test score, and here's a bunch of people who were sleep deprived. Their score looks like it's like 24 on average. Then here's a control group of people. They were not deprived of sleep. Well, they're doing, looks like 13, 14 points better, way up in 38 or so. The whole idea is people who are deprived of sleep are not performing at their peaks. And when it comes to final exams and things like that, you want to be performing at your peak, try to get good sleep, start your studying early. It turns out that if you interrupt sleep during stages 3 and 4, which are the deep sleep periods where you see the slow EEG waves, well, that will cause the same periods of slow wave sleep to be longer than average on the succeeding night. It's as though your body's trying to make up for the lost slow wave sleep on the preceding night. Ditto. If you interrupt sleep during rapid eye movement periods, then that will cause an increase in rapid eye movement sleep over the average on the next night. It's as though the body is trying to make up for the fact that it didn't have enough rapid eye movement sleep the night before. So your body's got to have adequate amounts of both forms of sleep, and if it doesn't, it will try to make it up on succeeding nights. Here's an important one. Several nights of partial sleep deprivation decrease the body's immune response. So all these people who are working too hard and not sleeping quite enough, they're going to go around and get colds, or maybe they'll lose their voices. How many people have a friend who has sort of lost their voice this winter? There are a whole bunch of people where I work. I see some of you also. Anyway, overwork and not enough sleep, that can cause this sort of thing. Stress, lack of sleep. You want to do all the normal things that your parents probably told you to do, like try to relax occasionally, eat well, and sleep well. It really helps. Turns out that how much sleep is needed depends not only on the person, it also depends on the age. And that's shown in this next one. So here we go. Here is a timeline over the human lifespan. So we have 1 through 15 days. That would be more or less a neonate, all the way out through 90 years, and that's an old person. So the vertical axis is showing total daily sleep in hours. And the first thing you should note is that when you are a tiny infant, well, you might be sleeping 16 hours of the day, and that is true. And when you wake up, you cry, you get fed, you look around, then you go back to sleep. And this large amount of sleep decreases over the lifespan, so that by the time we have a person of age 90, then the average number of hours of sleep is 6. So that's far fewer than what we had before. If we were to look in your age group, then you're somewhere between 18 and maybe 22 on average for undergraduates. So you're somewhere around here. You could say, well, the average looks to be about 8 hours, maybe a little bit more. Of those 8 hours, it looks as though almost 2 of them, certainly 1.5 of them, are spent dreaming, or at least in rapid eye movement sleep. And then the rest of it is spent in slow wave or deep sleep. Somebody like me, well, I'm 56 years old, so I'm way out here, 6 hours of sleep. Actually, I sleep a little bit more than 6. I'm more like 7. But, and you can see that basically, as you get older, less sleep is required. I'm not certain if required is the right word, but people sleep less on average as they age. So, why do people sleep? That's a very good question. You have to sort of recall that the sleep that you might lose on one night is something that the body tries to make up for on the next night and the night after that. So the body does try to sort of maintain adequate sleep levels, and that sort of leads you to think that, well, maybe sleep is important. So one primary idea is that our bodies repair themselves, restore substances used up while we're awake, and if this does not occur, then all sorts of bad things happen. So, for example, physical fatigue leads to longer periods of slow wave sleep, true, and so it may be the case that parts of the body that are affected by physical fatigue are replenished during those periods of slow wave sleep, and so if one is fatigued physically, well, you need more slow wave sleep. Now, people ask, well, what is being repaired is not quite clear. Are there byproducts of metabolic activity that take time to sort of pass out of the body? Perhaps growth hormones released during sleep promote body repair. Perhaps they do. Why aren't those growth hormones secreted during the day then, so that the body repairs itself while one's awake? Well, people don't really know for sure what's going on. But it seems a good idea that, yes, the body is repairing itself, getting itself ready for action the next day when one wakes up. Let's see. If you look at neurons, people think that neurons are probably resetting their activity levels for normal daytime wakeful activities and consolidating connections that may have been made during the day. For example, new memories, new pieces of knowledge require new connections in the brain. So what we have here are images of fruit fly brains made to glow, and brightness is effectively indicating synaptic activity in these pictures. And at the top, what we have are well-rested fruit fly brains. And you can see that the brightness it's present, but it's not nearly as bright as what we have below, which is showing the fruit fly brains in sleep-deprived fruit flies. So it looks as though neuronal synaptic activity in the sleep deprivation case is higher than in the normal case, and that might signal some difference in function as a result of sleep deprivation that is normally absent in the well-slept fruit fly. So, in this idea, then, sleep may allow neurons to calm down after a day's activity, and that seems reasonable. So, second, neural activity during both the slow wave and rapid eye movement sleep helps to consolidate memories. Now, this is an idea of long standing, and what you can do is effectively study a list of vocabulary words, pretend you're back in junior high school or something, and you can then follow your studying by a period where you're sleeping, followed by the exam, or you can follow your period of study by some period where you're awake and doing something else that's unrelated, and then you take the exam. It turns out that the people who sleep after they study are going to perform better on average than the people who go out and perform some other activity after they study, not sleep. So people think that sleep can help one consolidate memories, and that can help with studying vocabulary lists for foreign language classes, for example. Turns out also that performance on certain motor tasks improved markedly after a period of rapid eye movement sleep. Now, there is yet another idea about what sleep is good for, and at this point you should be saying to yourself, well, sleep might be good for more than one thing. It's often the case that something that is very important fulfills more than one role. It's good for more than one thing. So here's a sort of third thing it may well be good for. Organisms need time to digest food, and certainly you're probably aware by now that when you eat a big meal, well, there's a lot of blood in your digestive tract, and lots of activity down there. And in some sense, it takes away from the energy that your brain could be using to think or do other tasks. A lot of people, in fact, they'll eat a big meal and they'll sort of feel sleepy. It takes time and energy to digest food. And perhaps the best way to do that is to sleep during that process. And one could be sleeping sheltered and safe from predators. Another notion is that with daytime animals or creatures like ourselves, well, if we're conducting a lot of our important tasks using the sense of sight, then when it's nighttime, well, it's very difficult for us to be performing important tasks, and so we might as well be sleeping. And that, for me, is not a very compelling argument, because there's lots of nocturnal animals that see perfectly well at night and do a lot of their hunting and things like that. Why are we not that way? I don't know. But it's certainly the case that unless there's a campfire, in our natural state, humans are not getting a lot of light at nighttime. And so that makes sense then to sort of adopt an inactive state where one is sheltered, safe from predators. So sleep. Well, we now know there are periods during sleep of rapid eye movements, and during those periods our eyes are moving around underneath our eyelids. Our bodies are more or less paralyzed, so we don't start kicking and punching in bed and stuff like that. And this is typically the time when people have the most vivid dreams. And this is not to say that these are the only periods where people are dreaming. It's believed that people are dreaming during slow wave sleep, that these are typically less vivid and in some ways less memorable dreams than the rapid eye movement dreams. So, dreams are associated with rapid eye movement sleep, and they're often pictorial, so that we have visual imagery, and it seems more or less real in some sense. We're the actor, and something real is going on. Well, we know it's a dream, I guess, but we're certainly reacting to what's going on in the dream as though it were real. We're taking it seriously. I guess that's the way to say it. We're taking our dreams seriously. So it is the case, like I said, people do report dreaming if they're woken out of slow wave sleep, but these dreams are typically less vivid and engaging. If you wake somebody out of rapid eye movement sleep, then they'll typically report that, yes, they were having one of these vivid, more memorable dreams. So, interestingly, rapid eye movement sleep dreams have a reported duration that matches the duration of rapid eye movement sleep. So if you have 15 minutes of rapid eye movement sleep, you can have a 15-minute long dream. That's pretty interesting. Okay, dreams often include current daytime activities and emotional preoccupations. And I've noted a couple of examples of daytime activities. So there was a period where I said, I'm going to play Tetris. And when I went to sleep that night, I had little dreams of these falling blocks. And clearly, it was because I was trying to play Tetris during the day, and then I go to sleep and dream about little things falling down like this. It's really silly, but clearly, daytime preoccupations show up in our dreams. Emotional preoccupations, I think, also. But there are sort of complications there that I'll talk about. A further thing that dreams include are weird or illogical elements. So, how many people have ever flown in a dream? Flying is really neat. It's believed to be a slight vestibular system screw-up, a temporary thing, that causes a feeling that actually you're able to fly, and you start flying around in your dream, and it's really neat. So that's a good one, and that's definitely weird. Being naked in public, showing up to a final exam late. What are the other sorts of embarrassments or bad situations that people have in dreams? Well, I guess there's all sorts of them. being late for the final exam. Don't be late for the final exam. It's okay to have that in a dream, but not in real life. Recurrent themes for most people, well, illness, fighting, romance, and you can name a theme, and there's probably somebody who has recurrent dreams on that theme. I have recurrent dreams about being in weird buildings or, I guess, urban environments. I'm trying to find my way through these buildings. I'm on these streets. And that sort of thing occurs at least once a month. And I sit there, what is that all about? I have this feeling of, where am I? Feeling lost in this sort of weird, anyway. That's a recurrent kind of dream for me. I'm not certain what the theme is, but I know it's one of these constants for me. Now, you may discover certain things like that for yourselves as you get older. Relatively few dreams reported have detectable sexual content, interestingly enough. However, rapid eye movement sleep in both males and females is associated with genital arousal. So, even though there may be no sex in the dream, there may be genital arousal nonetheless. Now, here's one of the things most people have noted, and it's sort of a bummer. Most dreams have predominantly bad outcomes and negative emotions. Does that seem true? It seems sort of partially true to me. At least some of the most memorable dreams, something bad is happening and you almost want to wake up out of the dream so you can stop it. There are certainly pleasurable things in some dreams, like, when I'm flying in a dream, which is really rare, but when I am flying in a dream, that's really cool. You say, oh wow, finally I can fly. And you fly around and do things. But there's a lot of dreams where there are bad outcomes and negative emotions, and that's just the way it is. So you sort of have to grin and bear it. What's going on with dreams is the logical question for a psychologist. Well, many people propose that dreams have hidden meanings. And Sigmund Freud was one of the earliest developers of this notion. Dreams present latent or hidden content, and it consists of unconscious desires and impulses that are normally masked by self-censorship. So there are a number of things effectively that we are taught should not be done. So murder, adultery, all of the things in the Ten Commandments, just for example. These are things we're taught should not be done. And a lot of time if we encounter that thought while we're consciously aware and awake, we might say, well, let's not go there. However, this will come out when one's dreaming, because the idea is that, well, all of this latent content that we normally think about, that we would normally think about as humans, rises to the fore. All of these unconscious desires are still there, and while we might try to repress them in our daily lives, well, they come out at nighttime when we dream. That's the basic idea. A lot of people think that this is true. And in this particular graph, we've got people in the United States, people in South Korea, people in India, and they're effectively responding what their best theory concerning dreams is. And the Freudian theory is basically, an expression of this latent content arising from the unconscious desires. That's what Freudian means. Problem-solving dreams are there to help us solve problems. Dreams are there to help us learn. Dreams are sort of byproducts of other activities. And you can see that in all three locations, most people are reporting that, yes, they believe that dreams are expressing latent unconscious desires, the Freudian notion. Now, just because people think that doesn't make it true, but people tend to believe that on the whole. Now, what about some of those other things? Other researchers suggest that dreams are just a byproduct of other brain activities. And so, for example, what happens is that the pons in the brainstem here produce bursts of neural activity during dreams, and these activate neurons in lateral geniculate nucleus and in occipital cortex. Now, you know that if we are activating neurons in LGN and in occipital cortex, then we are going to end up seeing something, because these are neurons in our primary visual pathways. So effectively, as a result of these bursts of activity in these pontine neurons, we have vivid imagery, and it's thought that, well, this is actually producing what we're seeing during the vivid dreams present in rapid eye movement sleep. So you have to ask yourself, well, if it's the activity of these neurons here that are causing us to have vivid dreams, and in some sense, it's not really the expression of latent unconscious desires that dreams are all about. Rather, it's sort of more a byproduct of this neural activity down in this area of the brainstem. That's the way this argument goes. So these PGO waves result in vivid imagery. Now, you might just step back a moment and say, well, okay, so there is some reason why we have vivid imagery during rapid eye movement sleep. These neurons are active, so we get these PGO waves. But that doesn't mean that there aren't further areas of the brain which respond to this vivid imagery and try to make sense of it. And maybe it's these other areas of the brain that are expressing our latent unconscious desires. So let me continue with this, though. So priming by a person's recent experiences and recurrent thoughts brings certain images into awareness. So if we have been looking at little Tetris cubes falling all day, then if we have a dream at night, well, it may be that the activation of visual cortex in rapid eye movement sleep by these PGO waves is going to result in visual activity interpreted as little Tetris blocks falling down. A lot of time, the images that come to mind are not linked in a systematic way. There is no orderly narrative. And in fact, there does appear to be some random element when you think about some dreams and the kind of pictures that one gets. And that suggests, again, that, well, you're not really starting with a coherent story that you're telling. Rather, you may be responding to these random PGO waves generated by pontine neurons. And so a lot of people at this point think that, yes, there are portions of the brain involved with imagery and visual sensation that are stimulated during rapid eye movement sleep. And the dreamer, in particular, the other parts of the brain that are good at interpreting visual imagery, they attempt to synthesize these visual elements into a somewhat coherent narrative. And it is at this point that the latent unconscious desires can be expressed. So there's effectively all this random stuff coming up on the screen. We're very good, though, at interpreting stuff coming up on the screen. And how do we interpret it? Well, in terms of our latent unconscious desires. That's the idea. So, hypnosis. And if it appears like I'm sort of running through this rapidly, I sort of am. And the reason is I want to talk about the final exam also. So bear with me. We're now going to talk about hypnosis briefly. Then we'll talk about meditation briefly. And then drugs. We're getting there. We're getting close. Hypnosis. Well, everybody's seen examples of hypnosis, if not in real life, then in a movie probably. And hypnosis refers to a highly relaxed, suggestible state of mind in which a person is likely to feel that the actions and thoughts are happening to him or herself rather than under his or her control. I've seen so many movies with hypnosis at this point. So I remember there's this really old Sherlock Holmes movie, and there's a woman who has a bowl of water with little flowers. And the flowers are sitting on top of the water, and she starts stirring the water. You are getting sleepy. You are getting sleepy. And the guy is hypnotized, and then he ends up committing a murder, because it's a Sherlock Holmes story, of course. Anyway, you can probably think of your own examples of hypnosis. And the notion is that a person is asked to sit quietly and focus on some sound or object in the environment. And it may, in my example, be the swirling water with the flowers. The hypnotist would speak encouragement to the subject. Please relax. Your eyelids are getting droopy. And if the the subject is sufficiently suggestible, then yes, the person will enter a hypnotic state. Now, if you watch movies, you'll have probably some fixed notions about what can happen in a hypnotic state. For instance, a hypnosis will say, please go murder somebody, and you'll go out and murder them. Well, in a Sherlock Holmes movie, maybe that's what happens, but it's not really true. Basically, hypnotism does not cause you to do things you wouldn't otherwise do. So maybe if a murderer is hypnotized, then yes, they would go out and commit a murder, but normally speaking, that would not occur. So no worries. Hypnotism cannot return a person to an earlier age. So if somebody hypnotizes you to try to find out what your childhood memories are, that's not really happening. Sorry. You're getting your present day memories. And you might try to interpret these in terms of what you think a child would think, but it's still bogus. You're not regressing to an earlier age. Finally, there is no reason to believe that hypnosis improves memory. And in fact, courtroom testimony enhanced by hypnosis is banned. Let's just forget about it. It's bogus. Hypnosis, then, it's sort of hard to get people to do things with hypnosis on the whole, although there are some notable good examples that I'll talk about in a moment. It doesn't help a person think about earlier age stuff, and it doesn't help with memory. But suggestions as to how one should behave after hypnosis can be powerful. So for example, there's all sorts of hypnosis therapy concerning the cessation of bad habits. Chief among these is probably smoking cigarettes and whatnot. So people go to their hypnosis therapy. They're smoking a pack or two of cigarettes a day, and the hypnotist will hypnotize the person, tell them to stop smoking, and then they'll wake up, and a lot of these people will actually stop smoking. So there's some power to the therapy, the suggestion made under hypnosis. Other things that appear to work are suggestions to forget everything that happened during hypnosis. So if you ask a person to forget everything that happened during hypnosis, well, it's likely that they will. They will not be able to report what happened. So these are true facts concerning hypnosis. Some of the things on that previous slide, well, those are the made-up things that don't really happen. Here's another one, pain reduction. Pain, for some people, is something they have to live with. And it's really a critical issue how to reduce the pain. And so administering a placebo pill to a person with a lot of pain, on the whole, may have very little effect. Aspirin may have more. Here's morphine, which is pretty serious stuff, addictive. Acupuncture results in a fair amount of pain reduction. But look at this. A person who's hypnotized has a large amount of pain reduction. They are feeling no pain, relatively speaking. The effects are much greater than the administration of morphine, which is in fact a standard way to reduce pain, big time. So, algesia, A-L-G-E-S, that refers to pain. A-N is without. So we're talking about ways to be without pain when we're talking about analgesics. And one of the ways is to undergo hypnosis. It's possible to reduce pain substantially. So, how does hypnosis work? Some people would say, well, it's an exaggerated form of social influence, effectively one person bossing some other person around, to put it plainly. And in this case, the hypnotist has an enormous influence on a person hypnotized. Well, that's all very well and good. Yes, we would say that the person being hypnotized has to be fairly suggestible, and the hypnotist, him or herself, is going to be delivering instructions. So there's certainly some truth to the influence here. However, it doesn't explain some of these effects, the post-hypnotic effects or the hypnotic analgesia. So it's not a complete explanation. Another theory is that there is dissociation in a person's psyche. So that effectively during hypnosis, there's part of the person that stays with the body, the normal ego, I suppose, and then there's another aspect of the person that leaves that perspective and sort of floats around outside of oneself. Well, if you're floating around outside yourself, not directly attached to your body, that sort of says why you might not feel as much pain. It doesn't really help us understand why we should stop smoking or forget about what's happening under anesthesia, under hypnosis. But it's certainly an interesting idea. Meditation. People believe that certain forms of meditation, if practiced appropriately, can lead to alterations in consciousness. People talk about higher states of consciousness and things like that. And certainly this is one of the, and I'm probably going to express this incorrectly to any Buddhists out there, but certainly in Buddhism, achieving higher states of consciousness through various behaviors, in particular meditation, is one of the most important things one can do. And so we might ask ourselves, well, are there any changes in brain activity as a result of meditation? Are there, in fact, different states of consciousness that we can collect physical evidence about using brain imaging techniques? So, turns out that, well, brain activity changes during meditation, especially with those who have a lot of practice. And what you'll find are pronounced alpha rhythms, for one. And we know that alpha rhythms are normally associated with, well, relaxing and closing one's eyes and just sort of chilling out and stop worrying. That's a good way to have increased levels of alpha rhythm. And so this might be simply an indicator that a person who is meditating is in a more relaxed state than they would be otherwise. People also find more pronounced gamma rhythms. And if I haven't mentioned EEG gamma rhythms before, these are effectively maybe 30 hertz or above. So these are very rapid waves in the EEG signals. They're thought to be involved in coordinating and integrating brain activity from different brain areas. In any event, if you look at brain waves in a neutral state where a person is not meditating, compare that to somebody who is meditating, well, gee, there are differences in brain activity. And we have this plot from the textbook here that shows some of these changes. Now, whenever I see a plot like this, I'm afraid I'm going to have to be a little bit critical. We have a neutral state, we have a meditative state, we have an electrical activity. I wish the electrical activity had units, physical units of measurement, number one. Number two, when I see little points like this, this, and this, and this, well, I say those are pretty close. Those are a little bit farther away. I just wonder how close these data points are to one another. So what does one often find in a plot of data to help us understand what the spread in the measurements is? Error bars. When I look at this, I say, where the heck are the error bars? Are these two really different electrical activity values? Or are they just differences found, just as a result of chance? Well, the error bars would help me know whether there's actually a significant change from the neutral state to the meditative state. As it is, this plot is useless for me. So, how many people have taken their statistics course? How many people are looking forward to taking their statistics course? Because that will let you identify poor figures that do not provide sufficient information. In any event, people are interested in whether or not meditation changes brain activity, and there is now a fairly convincing set of evidence that it does. Greater alpha rhythms and increased gamma activity. We're nearly there, folks. The basic message at the end of chapter 6 is that if you take drugs, your brain will do funny things. If you take too many drugs, then you'll end up a vegetable, an addicted vegetable. So if you don't want to hear any more about this topic, well, just go back to junior high school and your DARE police officer telling you not to take drugs. That's effectively the end of this chapter. However, I will note that drugs, we have depressants like alcohol, sleeping medications, anti-anxiety medications. These all are depressants. Stimulants, on the other hand, include caffeine, cocaine, amphetamine, ecstasy, and these will decrease one's need for sleep and lift a person's apparent energy level. There can be some very bad side effects for some of these drugs, including addiction in a number of them. And there's also pictures like this. This is before meth, this is after meth. You've seen pictures like this before. Breaking Bad may be fashionable among some people, but don't take it literally. You don't want to do meth. Marijuana, it's legalized in a number of states. In fact, in this state, there are medical dispensaries where people purchase marijuana. So marijuana activates cannabinoid receptors, and this is something you'll learn about if you take a course in neuropharmacology. It activates cannabinoid receptors in the brain that normally respond to another substance called anandamide, and this is involved in mood and appetite regulation and in pain perception. So it turns out marijuana is also a very mild hallucinogen in the sense that one can have spurious visual imagery with a large dose of marijuana. There are far more powerful hallucinogens in marijuana, however, including, for instance, lysergic acid diethylamide, LSD, PCP, mescaline, what have you. While these drugs can produce powerful perceptual experiences through these hallucinations, note that they can also produce a lot of emotional distress and delusions, particularly if one is not prepared for the fact that, well, there's going to be a hallucinogenic world presented to one. So it's really more dangerous than one might think to take hallucinogens. What about drug addiction? Well, I can advise one to not be addicted to drugs. People have different susceptibilities. So I'm a former smoker of cigarettes, so I know about addiction, at least for something not super bad. And there are some people who are fairly easily addicted to substances like that, and then there are others who are not. And I think it depends on the actual substance. So for example, some people, if they have one cigarette, then that is typically enough to hook them for their entire life. Other people, they have one cigarette and they could care less, it has no effect. So there are certainly differences in tolerances and susceptibilities among individuals, and these are likely due to genetic factors, inherited, personality, as well as environmental factors. So if all of one's peers are doing a particular activity, well, I think one is more likely to do that, him or herself. Social support is also very helpful if somebody is trying to beat a form of addiction. So if other people are there saying that, yes, quitting a particular habit is a good thing, then that can be very helpful. That's the end of chapter 6 in my book. Thank you for bearing with me. Let's turn now to the final exam. And what do I need to do here? There we go. The bottom line is that the final exam is not all that hard, and it's very similar to the midterm exam. So I guess I should say, if you found the midterm exam really hard, you'll probably find the final exam really hard. But if you didn't mind the midterm exam too much, I don't think you'll mind the final at all, because it's more of the same. It's not super hard. I think most importantly, people have got to show up. When is the final exam? This coming Tuesday. What is today? Thursday, Friday, Saturday, Sunday, Monday. You have 4 days in which to study for this exam. Okay. March 18th, yes, 2014, 8 in the morning. So those of you in class today, and it's good to see you, well, you're here 8 in the morning. And just like the midterm, our final exam will be at 8 in the morning. If you show up at 9:30, well, you're only going to have a half hour to do that exam. And if you're a super fast whiz, you may well be able to complete the exam in a half hour, but generally you'll want more time. Show up a little bit earlier than 8 o'clock, just like for the midterm. I mean, you'll want to get your stuff prepared. You want to fill out your Scantron with your name and ID number. You'll want to see which form of the exam you have, blah, 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 blah. So it's always a good idea to show up maybe 15 minutes early or something when it comes to an exam of this sort. Now I see a hand.
It causes insomnia.
Say it again?
Insomnia.
What is what?
Yeah. What causes that?
It's often caused by stress. Yeah, I think. In my experience, if I have insomnia, it's often because I'm thinking about a stressful circumstance that I don't have a solution to. That's my personal experience. Now, there are people, I think, who have other forms of insomnia that are not the kind that I've experienced. I don't actually experience insomnia, but, you know, in rare. I think there are people who have more chronic insomnia who might take sleeping pills as a result of prescription from a doctor, and I don't really know why they have insomnia. So the answer is I don't know. It's probably something on a wiki page that you can look up pretty rapidly if you'd like to. Okay. Try to get a good sleep before the finals. Now, here we go. The final exam will cover textbook material from chapters 1, 2, 3, 4, 5, and 6. And 1, 2, and 3 are there. That means this is a cumulative final exam. But the emphasis will be on the second half of the course, chapters 4, 5, and 6. The final is worth 125 points. And at this stage, what I'd like to say is, well, what are the points for this course? Well, we now know the final exam has 125. What about the others? The midterm, that was 100 points. There are also 10 points for the bonus essay, so that's a total of 110. Then the final is worth 125 points. So what is that, 235? Then the final also has a bonus essay worth 10, so that's 245. There's 6 extra credits, 251. And then 25 points are added to everybody's score, and that gets us up to 276. So in fact, the total number of points offered in Psych 9A, PSB 11A is 276. Do people remember how the grades work? Well, it's pretty straightforward. There's no curve. It's simply 225 points or above, you're going to get an A of some sort. Somebody who is close to 225 or above will get an A minus. Somebody who is significantly higher than 225 will get an A, and somebody who gets a nearly perfect score will get an A plus. I don't give out very many A pluses. Now, 200 points or above is going to be some kind of B. Again, if you're close to 200, it's probably going to be a B minus. If you're significantly above 200, 210 or something like that, that's probably a B. And if you're close to 225, maybe 223 or something like that, that's a B plus. I actually figure out where to draw those lines when I'm looking at all of the scores that people have. But that's the general trend. Somebody has 175, well, they've gotten likely a C minus. Is C minus a passing grade? No, it is not considered a passing grade in many circumstances, oddly enough. If you want a passing grade, you want a C or better. You really want to get something like 180 or 185 to make certain you're getting a C rather than a C minus. Now, the people in this zone, you don't want to go there. I don't like giving bad grades. People don't like receiving bad grades, so you just don't want to go down in that low area. There were several people we noted on the midterm review who had really pretty poor midterm scores. And you probably know who you are if you're still in the course. Wow, I hope you pull up the grade on the final exam, because I don't want to give grades for this sort of thing. Okay. What are the questions on the final exam? It's really similar to the midterm. There's multiple choices, there's picture labelings, and then there's a bonus essay. How many multiple choice questions are there? 55. How many were there on the midterm? I think it was 35. So there's actually 20 more multiple choices on the final than there were on the midterm. So 55 multiple choice questions, each is worth 2 points. That's a total of 110 points. Then the picture labeling questions, actually there are fewer picture labeling questions in the final exam. I can tell you 2 things that you really got to study well for picture labeling. One of them is the eye, and one of them is the ear. And when I say the eye, not only do I talk about the parts of the eye that are visible, but also some of the more microscopic structures at the back of the eye. Okay? And we looked at lots of diagrams of the retina, for example. You want to look at that. What about the ear? Well, you want to know what the ear canal is called. You want to know what the pinna is called. You want to know about the eardrum and then the hammer, anvil, and stapes, those little bones. But you also want to know about some of the more microscopic structures found in the cochlea. So when I say ear, I don't mean just the big bits. I mean the microscopic bits as well. Anyway, picture labeling questions. There may be more pictures, but those are the 2 primary things to study. 15 points total. And if you add up 110 for multiple choice and 15 for picture labeling, hey, that's the final exam score of 125. Now, there's a bonus essay. That's 10 extra points. And I'll tell you what that essay is shortly. So why is this exam cumulative? About 1 out of every 5 multiple choice questions comes from the earlier chapters 1, 2, and 3. So 80% are from the late chapters 4, 5, and 6. 20% are from the earlier chapters 1, 2, and 3. And that's just to keep you on your toes. In fact, those of you who remember the sort of questions you had on the midterm may find very similar questions on the final for that cumulative section, the 20%. Okay. What would I be studying if I were in your shoes? Well, I would be watching videos of these lectures together with the PowerPoints, because you'll know the sorts of things then that I emphasize and find important in our textbook. So I'd certainly do all of that. And the lectures are shown here, how they're broken down into chapter. And your emphasis for the final will certainly be these lectures in the second half of the course concerning chapters 4, chapters 5, and chapter 6. But if you have the time, look at the early ones also. Second, everything is really coming from the textbook in this class. That means you've got to be reading the textbook yet again for the highest possible performance. So try to look over the lectures and try to look over your textbook. If you find stuff in the lectures that's not in the textbook, don't worry about it. I sometimes try to add things to what the textbook is saying. The final exam, though, will be focusing on material that you can find almost always in both locations, the lecture and the textbook. We'll talk about this. So the textbook is where things come from, ultimately. So, you know this stuff already. Bring one of those Scantrons. Bring some pencils. Bring your photo ID card. If you forget to bring your photo ID card, don't freak out, because I have a way to find your picture through the registrar. But I'd prefer not to do that. So bring your photo ID card. Same thing as before. No books, notes, calculators, or electronic devices. Everything will be stowed under the seat. There's no need for a blue book. There will be a bonus essay, and you can write it on the page in the exam itself. So I'm going to give you some sample multiple choice questions, and it's just like the midterm. Choose the best answer. You might say, well, I don't really think any of these answers is perfect. And in fact, maybe not, there's not a single answer that is perfect. However, there's almost always one that's sort of head and shoulders above the others. And that's the one you should choose. Choose the best one. Not the perfect one, but the best one. Second, if you find a good one that you like and that you don't read down the list of all possible answers, you might miss a better answer at the bottom that you never got to. So really you've got to read all of the answers to make sure you do not choose the second best. Finally, I didn't write it here, but I'm going to say it out loud. There is no penalty for guessing. Should you fill in each answer? Each question? You certainly should. I mean, you might not know the answer, but your guess might nevertheless get you some points. So answer each question because there is no penalty for guessing. Okay, here are some sample questions. These are drawn from the latter half of the course, chapters 4, 5, and 6. So, when colored lights are combined, one uses the rules of to understand the resulting color. When color pigments or filters are combined, one uses the rules of I actually know the answer right off the bat, and that's because I've done a lot of color vision research. But if you don't know the answers right off the bat, probably the best procedure is to insert the words in each of the possible answers and see if they make sense. So let's try A. When colored lights are combined, one uses the rules of Young-Helmholtz theory. Well, what I recall about Young-Helmholtz theory is that it says that color vision is trichromatic, and that there are 3 types of color sensor. So we have a 3-dimensional color experience. That doesn't seem to make sense here. Let's try the second half. When color pigments or filters are combined, one uses the rules of Herring color opponent theory. No, that effectively is talking about how we combine photoreceptor responses, not how we combine pigments or filters. So I would say A is not a strong answer. How about B? One uses the rules of trichromatic vision when you're adding lights. You're using dichromatic vision for color. What is dichromatic vision? Well, this is typically what a person has if they have severe color blindness. They are totally missing one kind of photoreceptor, which is often either the long wavelength sensitive photoreceptors or the medium wavelength sensitive photoreceptors. And as a result of this dichromacy, that person is going to have red-green color blindness. So effectively, somebody who is a dichromat has 2 dimensions of color vision, and that's because they're missing one of the normal 3rd dimensions, and that's typically red or green. So that doesn't make any sense whatsoever for this particular answer. Okay, what about C? Well, C is our answer. If you are mixing lights, then you use the rules of additive color mixture to understand the resulting color. And so, for example, if you have a red light, a green light, and a blue light, and you shine them all at the same location, it's possible to have a white light as a result. You will never get a white light if you're mixing pigments. So if I have a red crayon and a green crayon and a blue crayon all at the same location, I'm going to get this ugly dark mess. It's not going to be white at all, okay? Because those crayons are involving the mixture of pigments, and they follow the rules of subtractive color mixture. And that's this one. So C is it, but don't be confident to the level of excluding answer D. Let's try D. When colored lights are combined, one uses the rules of negative afterimages. What was our example in class where we had a negative afterimage? It was the flag. Do people remember the flag in the lecture? The flag gave us a nice negative afterimage, but it doesn't have much to do with combining colored lights. Color contrast, we also had some examples of that. That didn't have much to do directly with color pigments or filters, so that's not the right answer. The best answer, I think most people would say, is C. Okay, the receptive field of a cell. Well, I hope you'll recall some examples of cell receptive fields. Neuron receptive fields in the visual system, for example, we talked about these center surround cells that exhibit lateral inhibition. And we had examples where if we shine a light in the central zone of a cell's receptive field, then we can increase the cell's firing rate. But if we shine a light in sort of this donut-shaped surround, then we can end up inhibiting that cell's firing rate. So that cell's receptive field then comprises both the excitatory center and the inhibitory surround. And that's the example I have in my mind. And if we use an example like that, then what is the receptive field of a cell? Well, A, the total area of a receptor surface, the stimulation of which leads to an increase of that cell's firing. Is that going to work for our center surround cell? No, because it ignores the inhibitory surround that leads to the decrease in the cell's firing. So this is not perfect. What about this one? The total area, the stimulation of which leads to a decrease of that cell's firing. Well, for our example, that's not a good answer either because, well, hey, it takes into account the inhibitory zone in the surround, but it forgets about the excitatory center. If we're talking about a receptive field, we want to know about all regions that can lead to either excitation or inhibition. And that's answer C. The total area of a receptor surface is stimulation which leads to either a decrease or an increase in that cell's firing. So we're interested in how to change that cell's firing, no matter whether it's an increase or a decrease. And that will show us effectively what the extent of that receptive field is. That's answer C. So that's a good answer. And D, the area on the receptive surface which represents the transition zone from stimulation to excitation. That doesn't sound right to me. Stimulation and transition zone. No, it's definitely C. You want to include things that will lead to some change in the cell's behavior, and that could be a decrease or an increase in firing. So you want the center and the surround in our example. Distant objects produce smaller retinal images. And in class, I've been pointing out that, well, somebody sitting in the front row here, you're bigger than my thumb. People in the back row, though, well, you're smaller than my thumbnail. It's not as though you have different sizes in the real world, but the retinal image size changes dramatically as a function of distance. So, this fact is the basis for which of the following depth cues? Who wants to take a stab at this? Yeah, it turns out it's all of the above. Now, linear perspective. It turns out that if you have lines which are parallel in the real three-dimensional world, then they will converge towards a vanishing point. That means the lines, when they are close to you, look as though they are farther away in the picture. The lines, when they're now moving towards the vanishing point, towards a greater distance, they're now getting closer to one another in the picture. And again, the reason they're getting closer to one another is that same real three-dimensional distance between the lines results in a smaller retinal image as one moves away. And so they're getting closer as they move towards the vanishing point. So yes, linear perspective is certainly caused by this fact. Relative size. Well, that's more or less where you know the true size of an object, and you can use that knowledge to help estimate how far away the object is. So again, my typical example would be people. Adults have more or less the same size, at least globally speaking. So I would look at the retinal image size of this adult human right here, and I'd say, well, that is a pretty close person. I would look at the retinal image size of that adult human way back there, and I say, well, given the tiny retinal image size, it must be a person who's pretty far away. So, yes, relative size is intimately connected to this fact. Texture gradients, well, I hope you'll recall the texture elements, when they're close up, take a relatively large amount of the visual field. Texture elements, as they recede away from us, take a far smaller part of the visual field. And so that we can use texture gradients to indicate three-dimensional shapes. Do people remember those pictures in lecture? You might want to refer to those pictures in lecture to recall texture gradients. So yes, it's all of the above. And that is the correct answer. So, one effective orienting movements. And for me, the standard example is somebody over there says something and I go like this. I effectively turn my eyes and look in the direction where they're able to pick up maximum information, the direction of interest, and I turn my ears in that direction also, and that helps me perform more precise spatial localization and other things concerning hearing. So physical orienting movements help us obtain the maximum possible info. Which answer says that? Well, you know me, I like to read all the answers. So A, inhibit those sensory systems. No. We're not inhibiting sensory systems. We're trying to get the most information. That is B, so I think you're right. But let's check. Position the visual stimulus on the peripheral parts of the retina. No, in fact, we're trying to get the visual information centered on the fovea, where we have the highest resolution. Do we inhibit eye movements? No, we don't. In fact, when we're looking at something, typically our eyes are moving around. What are those fast eye movements called? Do you remember? Saccades. Yeah, so you're always making saccades, and when you shift to look at something, well, that doesn't mean you stop making saccades. You might fixate momentarily, but saccades continue. B. Descartes proposed that mind and body influence one another through, well, we actually had a tiny little video on this one. The answer is really strange. It's the pineal gland. So he thought that the pineal gland was effectively the way that the mind and body communicate with one another. So reciprocal consciousness. Remember, sometimes I make up new terms for you on the exam just to see if you can cut through the BS. I made that up. That is pure BS. What we would now term parallel universes. Well, it's an interesting thing, but it's not the right answer. The pineal gland is the right answer. Unconscious attributions are things that we do. We talked about that in the last lecture, but that's not correct for this one. It's got to be C, the pineal gland. Picture labeling questions focus on the eye and the ear, both the macroscopic and the microscopic structures. And here, finally, is our bonus essay question. Now, it says essay, but anybody who writes an essay is making a big error. I don't want to see any complete sentences. I don't want to see any periods. I want to see something that resembles more a table, and the reason is the more concise and systematic you present your answer, the quicker we'll be able to grade these questions. So, what is the question? Describe 5 Gestalt grouping principles. For each, please provide a description with 2 parts, its name and a little picture that illustrates it. So I want to see name, picture, name, picture, name, picture, name, picture, name, picture. Done. And if anybody starts writing whole sentences, I'm going to take off points. Just kidding, I won't take off points. You can write whole sentences, you can write whole essays if you like, but all I'm looking for is like a word or two that is the name of the grouping principle, followed by a little picture that sort of illustrates it. Now, what were some of those grouping principles? We had a list of these in lecture, and I think we started with things that are close to one another get grouped together. And what was that principle called? Proximity. And the example I had were 3 pairs of dots. And the dots looked as though they're organized into 3 pairs. And the pairing is in accordance with which dots are close to one another. That was my example. What's the second grouping principle? Things that are similar to one another get grouped together. So the picture in class was, well, we had the red dots and the green dots. And sometimes the reds and greens made columns, sometimes the reds and greens made rows. And it was clear that the similarity in color defined whether we perceived rows or columns. That was similarity. Things that move along a path smoothly are more likely to lead to proper interpretations than things that sort of have sharp corners. There's a sort of good continuation principle saying that if we have things that are intersecting, then we're likely to see, well, this as 2 lines that are crossing one another rather than 2 V's that just happen to intersect at the same vertex. So that's good continuation. What's another one of these grouping principles? Common fate is one that I like. Things that move together get grouped together. So if you have birds going this way and birds going that way, you will definitely see two groups of birds grouped by their motions. Well, you can put some dots, and some of the dots have arrows going to the right. Draw some other dots, they have little arrows pointing to the left. It's very hard to make a drawing of a movie. I know what you mean, but that would be one way to indicate it. Now, what's a fifth one of these? Oh, I know what comes to my mind. It is a triangle with contours that have been erased. And in fact, they're partially hidden by an occluding object, such as a rectangle. So my memory says, why don't we have the principle of closure? So if we see the top part of a triangle and the bottom part of a triangle and they're perfectly aligned with one another, well, let's close up the gaps. And that's the principle of closure. Why might those gaps be there in the first place? Well, there might be some object in front of the triangle blocking our view where those gaps are present. So occlusion, if it is a reasonable interpretation of what we're seeing, that's often why we have closure working, perceptually speaking. So those are certainly the 5 things I would choose. And again, it's word, picture, word, picture. There's going to be 10 things on that page, and that corresponds to 10 points. I'd like to thank all of you for being good students, actually showing up to lecture at 8 in the morning. And I wish you the best of luck for the final exam. So, see you.